All right, Ninja Nerds, in this video we are going to talk about the histology of the liver. If you guys haven't already, go see our video on the vascular supply or the circulation of the liver, as well as the portal hypertension video. We go into more of the gross anatomy on the liver, we talk about the blood supply to the liver, and then what we did is we kind of finished off that video talking about what's called a liver lobule. And that's what we're going to try to focus on a lot in this video, is we're going to really zoom in into the structure of the liver and to look at it at the actual tissue and cellular level. All right, and then after that we'll have another video and we'll focus at it specifically at the cellular level. All right, so let's go ahead and get started. All right guys, so now what we're going to do is we're going to really zoom into the liver, okay? There's this special structure that we're going to talk about. We're going to really zoom into it. If you guys remember at the end of the video, we kind of, around the time where we were talking about the liver circulation, we were talking about portal hypertension, we talked a lot about the blood flow to the liver. Remember, we talked a lot about the blood supply. And we said the main blood supply, we're not going to go into a lot of detail because we've already covered it, but the main blood supply to the liver was coming from the hepatic artery proper, right? So it was this artery right here called the hepatic artery proper. And we said that the hepatic artery proper was important because what it was responsible for doing is it's responsible for supplying oxygen-rich blood to the liver, okay? Supplying a lot of oxygen-rich blood. Then we said that there was another one. So again, before we spin that next one, what was this one right here called? It was called the hepatic artery proper. Okay? Then after that, we had another guy. And that one was really important because we said that that was applying 75% of the blood flow to the liver, right? Of the blood supply, right? And that was coming from your gastrointestinal tract. And this one was called your portal vein. If you remember, the portal vein was bringing blood from a bunch of different structures from your gastrointestinal tract. And that portal vein, this is your portal vein, it's bringing nutrient-rich blood. Okay, nutrient-rich blood, it might even contain toxins, it might contain bacteria, it might contain drugs, it might contain alcohol, a lot of different things, but the whole important thing is that this is providing 75% of the blood supply to the liver, 75%, and then the hepatic artery proper is only supplying a very small, minimal amount, that being approximately 25% of the blood supply to the liver. All right, now we said that there was another really important structure that was coming into the liver, or specifically, I should actually be more specific, draining the liver. Remember, there's these special structures here that drain the liver, and they contain a lot of bile. Right? They contain bile, and bile is important for emulsification of lipids. It's also important because a lot of the things that we actually, our liver metabolizes, if it doesn't want it in the body, can actually excrete it out of the body in the bile. So that's important. So this structure here, we said, is the bile duct. And we'll go over the biliary system in an individual video. For right now, I just want you to know that this is the significance here. So there's three specific structures that are coming through that porta hepatis. The ones that are going in to the liver through the porta hepatis is the hepatic artery proper and the portal vein, okay? The one that's coming out of the porta hepatis is the bile duct. Specifically, it's going to be the common hepatic duct, okay? Now here's the next thing I want you to understand. These aren't the only things that are going through that porta hepatis. We talked very briefly, and we said that there's also going to be a nerve plexus, okay, that's coming in to the liver. And if you remember, we called it the hepatic plexus. Okay, so it's going to actually come into the liver as well. So what is this plexus here called? They call it the hepatic plexus. And the hepatic plexus is really important because if you remember, the hepatic plexus is actually made up of sympathetic and parasympathetic nerve fibers. The sympathetic nerve fibers of the hepatic plexus is coming from the celiac ganglion. And then the parasympathetic fibers is coming from the vagus nerve, if you guys remember that. So again, that's going to be another nerve pathway. Last one, and that's actually going to be draining the liver. So there's actually three structures going into the liver, two structures coming out of the liver. And this last one here is actually called your lymphatic vessels. So one more that's actually coming out of the liver here is actually called your lymphatic vessels. These are important because they're draining the interstitial fluid, and that's important to be able to make sure that that interstitial fluid, any of the fluid that leaked out of the capillaries, these sinusoidal capillaries, is returned back to the circulation. That's the significance of the lymphatics. So this lymphatic vessels, these are going to be our lymphatics, if you guys really want to know, we'll talk about it more in the lymphatic system, but these lymphatics from the liver will actually drain into a specific nodes, like specific lymph nodes, and these are going to be like your celiac lymph nodes. Okay, so they'll drain into the celiac lymph nodes. So now there's an important thing that we should understand. All of these things, the hepatic plexus, the hepatic artery proper, the portal vein, the bile duct, and the lymphatics that are going to drain into your celiac lymph nodes, all of this structure that we consider to be. Here, I'm going to kind of like encase it like this. Let's do it like that. That whole thing right there, we actually call that a specific thing, and we call that the portal tract. Okay, so that's important to know. And remember, 3 things are going into the liver via the portal hepatis: hepatic plexus, hepatic artery proper, portal vein. 2 things are draining it: bile duct and the lymphatics. Okay, now one more thing that I want to do here before I keep going on, and I just want to talk about it very, very quickly, is if you guys remember, the liver is actually, we talked about this in the development video, the liver is what's called intraperitoneal. It's an intraperitoneal organ. What does that mean? That means that if you look at the liver, let's say here's the liver, okay, and here's your falciform ligament right there. If you look at the liver, it has a tissue that's actually covering its surface for the most part. And we call it the visceral peritoneum, right? We call it the visceral peritoneum. Now, the visceral peritoneum is then going to be connected to the parietal peritoneum. So now we're going to have another one, which is going to be kind of lining the wall. And let's just put it like this right here. Just like this for right now. Here is going to be the parietal peritoneum. Okay, so now we're going to have this right here, which is going to be called the parietal peritoneum. Okay? The liver is held inside of this peritoneal cavity by different types of mesenteries, like the lesser omentum, or the falciform ligament, or the other different ligaments that it has associated with it. But now, one more thing that I want you guys to understand. We know that it's actually going to be an intraperitoneal organ. Therefore, it has a mesentery, and it's covered by this mesothelial type of tissue. But one more thing. The liver has a connective tissue that's covering it. Okay, so it has this connective tissue that's actually covering it. And that's important. There's a connective tissue covering it, and that connective tissue that covers the liver is called Glistens Capsule. The reason why I want to let you guys know this is that Glistens Capsule is important because it's a connective tissue and it's actually under innervation, neural innervation, particularly from the intercostal nerves. So it has innervation by the intercostal nerves. Why is that significant? So when we talk about the nerve supply to the liver, there's not just the hepatic plexus, but there's also the intercostal nerve supplying the Glistens Capsule. Reason why is, let's say for whatever reason, the liver is inflamed. It's starting to distend. If it distends, it can activate the intercostal nerves in the Glistens Capsule, which can cause us to feel or sense pain. So that's important. It's an important concept there, is that whenever there is the distension of the actual Glistens capsule or distension of the liver due to hepatitis, cirrhosis, whatever it might be, whatever the underlying etiology is, it can activate these intercostal nerves, and that is important because it can actually relay the pain aspect of it. Okay? All right, cool. So I think we laid down a perfect groundwork for that. So now what I'm going to do is I'm going to take this liver lobule and we're just going to zoom in on it. And we're going to talk about all the structures associated with it. Okay? So first things first, let's talk about these 3 structures that you see on each end of this hexagon, okay? On this liver lobule, if you will. Okay, so this is our liver lobule. Our liver lobule, and again, why is it important? Because the liver lobule is the structural and the functional unit of the liver, okay? Now, what are these 3 things? We already talked about a lot of detail already. Remember this artery, the arterial, the hepatic artery proper? The hepatic artery proper will branch out into the right and left hepatic arteries and then into the arterioles. Those arterioles will go to these liver lobules. So what is this right here called? This right here is going to be the hepatic artery. This is going to be an hepatic arterial. Okay, so that's a hepatic arterial. Then, if you look over here, guess what this is going to be? That's going to be a tributary of the portal vein. So then this is going to be a portal venial, okay, which is going to be a tributary of the hepatic portal vein. And again, that branches onto the different parts of the liver. And then the other one is going to be your bile duct. Okay, it's going to be the bile duct. And again, what you're going to notice is that these three structures are located on every point of this hexagonal structure. Now, when you look under a microscope, you're going to see these structures, okay? And it has a particular name, if you will. These particular structures, if I encase these guys like this, have a particular name, and this name is called the portal triad. Okay, it's called the portal triad. Now, the reason why I say that is because what else did I tell you was technically at that area? Well, there's also going to be neural innervation, and there's going to be lymphatics. You can't see this next one. I'm going to put it outside of it, just for the sake of this situation here. But this is there. And what was that? That is going to be a lymphatic vessel. So this is going to be a lymphatic vessel. So now, here's why this is important. Let's say that if I were to actually understand under histological, I can't really see the lymphatic vessels when I'm looking under the microscope. But I should know that this structure is still there. Now I call this entire structure the portal tetrad. Okay, so make sure you understand that there is technically a portal tetrad, but again, when you look under a microscope, it's super, super hard to see the lymphatic vessels because they're blinded. They're very, very tiny. So we generally call this the portal triad. Okay, but do realize there's lymphatic vessels there. Now, next thing that we should understand is we know what's at these portal triads. Next thing I want you guys to remember is we said that the arterial and the venial are dropping things off to the liver, and the bile duct and the lymphatics are draining it. Let's see exactly how the hepatic arterial and the portal venial is doing that. At each end, you're going to see this like red, this kind of maroon structure here. This maroon structure here, I'm going to kind of put a little lumen here inside of it. I'm going to have a little lumen here. And that right there is the lumen by which, guess what? The hepatic arterial is going to drop its blood off into. And guess what else? The portal venial is going to drop its blood into this. And now we're going to have a mixture of what? The hepatic arterial blood, which is going to be oxygen-rich blood, and we'll have a mixture of the actual portal venial blood, which is going to be nutrient-rich, might be rich in bacteria, pathogens of different types, right? But that's the significance that I want you guys to understand. And what's going to happen is this blood is going to move centripetally, right? It's going to move towards the center where this blue structure is. And this is going to happen from each edge. So if you were to imagine, it would actually drain here, move towards that central vein. They would both drain into this sinusoidal capillary, move towards this one, and then same vice versa for all of them, okay? Now, because this blood is mixing and it's moving towards this vein in the center, we call this vein right here, smack dab in the middle of this liver lobule, we call it the central vein. Now, the central vein is important because the central veins from these different liver lobules will come together. So if you imagine for a second, let's do it like this. Let's say for just a quick second, let's say here I have a central vein of a liver lobule, okay, right here. So there's that central vein. I have another central vein. That one comes up. These central veins can connect together and form another structure. So this is my central, I'm going to put CV for central vein. Then they're going to come together and they're going to form what's called an interlobular vein. And then those interlobular veins will eventually come together, multiple of them, and it'll form eventually your hepatic veins. And then the hepatic veins will empty into the inferior vena cava. So I just want you to understand the relationship of this, okay? So these guys are dropping off blood into these sinusoidal capillaries, these little maroon things here, and the blood is mixing and moving towards the central vein. Realize that from these central veins, a bunch of them can come together and form interlobular veins, which can come together and form hepatic veins, and then hepatic veins can come together and empty into the inferior vena cava. So that blood is going somewhere, okay? It's not just going there and stopping. All right, so now, I've already mentioned this about a couple times. What are these maroon structures that I said? These maroon structures, this one here, there, all these guys, they're specialized capillaries that we talked about in the cardiovascular system videos. And these are called, they just denote them as sinusoids. Okay, but sinusoids are special types of capillaries. There's 3 types of capillaries, if you guys remember. There's continuous, there's fenestrated, and then there's sinusoidal capillaries. Out of those, the most permeable is the sinusoidal capillaries. If you remember, we said that they have a lot of fenestrations, little pores inside of the endothelial cells, and they have large intercellular clefts, which allows for a lot of macromolecules and plasma and solutes to move in and out of the blood vessel and into the interstitial spaces. That's important. And if you remember, we said the sinusoidal capillaries can be found in very specific places in the body. 3 of those important places, we said, is the liver, the red bone marrow, and the spleen. And the reason why sinusoidal capillaries are really good at trapping old age and defective red blood cells. And that's important because you're going to see some special cells in these sinusoids called Kupffer cells that play an important role in that red blood cell degradation. All right, so that's good enough right now. So what do we know? We know that these guys are dropping blood up into the sinusoids, and the sinusoids are taking the blood towards the central vein. Okay, that's good for right now. Let's do the next thing, the bile ducts. The bile ducts are really, really cool because they're picking up bile that are coming in between these actual hepatocytes. You see these cells here, these black cells? They're the ones that make up about 80% of the liver parenchyma, the liver cells, right? It's actually going to be your hepatocytes. These are your liver cells, and this makes up 80% of the liver parenchyma. Okay, that's insane. Okay. There's other cells that we'll talk about too, a part of the reticular endothelial system, like the Kupffer cells. We'll talk about stellate cells, but these are the most abundant and they make up most of the liver. And these are the ones that we're going to focus on in the next video. Okay. In between these guys, generally in between their apical membranes, and we'll zoom in on it after, I just want to get the basic thing here, is in between them you can kind of see this green stuff. That's called bile. The bile is actually secreted in between the apical membranes of these guys, where the hepatocytes are juxtaposed. There's a little apical membrane which fuses, but it forms a little small structure called a bile canaliculus. And what happens is the bile canaliculi come through these hepatocytes down between their apical membranes and then empty and drain into that bile duct. Okay? So now, what are these green things here called again? And we'll zoom in on it later, but they're called your bile or your biliary canaliculi. Okay? And we'll talk about those a little bit later. But now we understand that. Then there's one other area. And we'll zoom on that after. But you look here, okay, there's the sinusoidal capillaries. I'm going to highlight this in orange. In between the sinusoidal capillaries and these hepatocytes, you're going to see this space. Okay, if you guys can see that, that's called the perisinusoidal space, or another space that we're going to talk about called the space of Dissy. Any of the fluid that's being exchanged between these sinusoidal capillaries, we can't just let it sit in that tissue space. Okay, we have to drain it. So guess what? That can drain into these lymphatics. So the next one that I want you to understand is in between these sinusoidal capillaries right here is a nice little space. And what is that space there called? That space is called the space of Dissy. Okay? So we'll call this one the space of Dissy, or the perisinusoidal space. Okay? So now we have covered a big chunk of this liver lobule now. So now what I want to do is, so that we can really, really understand and even zoom in on some other structures that make up the liver lobule, is what I'm going to do is, imagine here is this sinusoidal capillary. And here's some hepatocytes right here. Okay? And then here's the portal triad. I'm going to take that chunk right there and blow it up. Okay? And we're going to look at it. Okay, so now we're zooming in on it, right? So just to get a little bit of orientation, again, this right here, you guys already know what this is our central vein. Okay, so this is going to be our central vein. And again, if you guys remember, they come together, interlobular veins come together, make hepatic veins empty into the inferior vena cava, which drains the liver and takes it up to the right atrium, as we know. Other things that you guys should understand before we really get into this is these little red structures here, these little cells that are making up this capillary. Again, what is this capillary here called? It is called a sinusoidal sinusoidal capillary. And again, just to review, what are the three places you can find this sinusoidal capillary? You can find it obviously in the liver, you can find it in the spleen, and you can find it in the red bone marrow. And why is it so special? If you guys remember, we said that it's really good at trapping old age and defective red blood cells that have reached their prime or they've reached the end of their prime, right, at 100 to 120 days. Okay, so that's our sinusoids. Then, if we look, we have this space in here. What is this space? It's that orange space. If I were to kind of highlight it a little bit in here, right, all this right here that we kind of highlight over there as orange, which is drained by the lymphatics. This right here is called the space of Dissy, right, or the perisinusoidal space. And then the next thing is just to review this. This is our actual bile duct. This right here, this green structure, is going to be called the Bile Canaliculi. And then we have this structure here, which is going to be our portal vein, which is a tributary of it. So this is our portal venule. So portal venule. And then we got this bad boy up here, which is going to be the actual hepatic arterial. So this is our hepatic arterial. And remember, this guy is giving the oxygen-rich blood and putting that into these actual sinusoidal capillaries. Two more structures that we get to finish off, and then we can really dive into this, is going to be this guy. This guy is so darn cool. He is called our Kupffer cells. Okay, these are called Kupffer cells. They're macrophages. They're a part of the reticuloendothelial system. They make up a large, large portion of it. Another one is going to be these like special brown cells here. They'd be here. They're found inside. Like for example, here's another one. This guy right here is called your hepatic stellate cells. They have two names. You can call them stellate cells, or another name that they have in textbooks is called the Ito cells. Okay? They're really, really interesting cells, and we'll talk about them too. All right, so now, last thing is we have these black cells, and these black cells are our hepatocytes. Okay? All right, so now, the blood, if you remember, from the portal venule is moving into this actual sinusoidal capillary, and the blood from this hepatic arterial is emptying into the sinusoidal capillary. And it's moving, right, centripetal as it's moving towards the actual central vein. We know that there's substances that are going to be in this portal venule blood and substances in this hepatic arterial that these hepatocytes are going to utilize. For example, if we were to zoom in on this, the blood flow going through there, you're going to have some red blood cells. All right, so let's say here is your red blood cell. In your red blood cell, you're going to have a special protein, which is called hemoglobin. And he's going to be in the oxy form, HbO2. We know that this is a systemic arterial blood. So the percentage of this oxyhemoglobin is like 98%. It's really, really saturated with the oxygen. What do we know? We know that this hemoglobin is going to let go of this oxygen, and it's going to deliver the oxygen to these tissue cells. Because these hepatocytes, man, they are hardcore. They perform a lot of functions, and they're unbelievable. All right, so these guys are really important. So the hemoglobin is going to be dropped off to these tissue cells, because your tissue cells, you know that they're going to be producing a lot of CO2. CO2, when combined with water, makes carbonic acid, and then that breaks down, right? So if we know this, you know your tissue cells are making a lot of CO2 combined with water. They make carbonic acid. Combined with that, they make what's called CO2. They make what's called bicarbonate. So bicarbonate. And they make what's called protons. And we know that the protons are important because they bind onto hemoglobin, triggering the Bohr effect to let go of the oxygen. Same thing is we know that a lot of the CO2 will actually bind onto hemoglobin and form the carbaminohemoglobin. And again, that'll all support to dropping off the oxygen to the tissue cells. So we know his significance is dropping off oxygen to the tissue cells. The portal venule, he's delivering a lot of blood that's rich in nutrients. So this is going to be tons and tons and tons of nutrients. So if you remember, and we'll talk about this more and when we get into the hepatocyte video, we're going to have a function of it. A lot of these nutrients are going to be taken up by these hepatocytes and used for various purposes, and we'll talk about that. But here's what I wanted to talk about. Other things that are unfortunately in the blood when we ingest food, and sometimes it can actually come from our actual intestinal lumen, is bacteria. So let's make bacteria, let's make it this blue color here. So let's say here we have this bacteria. This bacteria is not supposed to be here. We don't want it there. If it's there, we do not want this to continue to just funnel right past these sinusoidal capillaries, go into the central vein, and then get into our systemic circulation. That's bad. All right, because bacteremia, if in high levels, can cause septicemia. So we don't want that. We want to make sure that this bacteria is taken care of and we fix it before it leaves and goes into the systemic circulation. So Kupffer cells be thankful for them because they stop it right there. So one of the best things about these Kupffer cells, they're macrophages. What do you guys know about macrophages? They're phagocytes. So they love to engulf particle matter. So what will they do? They'll actually form those little pseudopods, right? If you guys remember, if you guys watched our immune system videos, they'll form those like little pseudopods and it'll ingest that actual bacteria. It'll take it in, engulf it. And then if you remember, if we take it inside, so let's say that we actually phagocytose it, internalizes this actual bacteria, takes it inside, and it puts it into what's called a phagosome. Okay, so if you remember here, we'll have like a little vesicle in there, and that vesicle will have the bacteria. Well, from this, it can do a bunch of different things. It can combine it with lysosomes. Right? So we could take this and combine it with special organelles that are present inside of this actual Kupffer cell, like lysosomes. When these two fuse, they form what's called a phagolysosome. And what does that mean? That means I can take this bacteria, okay, and I can start breaking it down, because what's going to happen is the lysozymes contain very, very powerful hydrolytic enzymes that'll start destroying this bacteria. And so it's going to break down this bacteria pretty darn good. Okay? So that's one of the beautiful things about these Kupffer cells, is that they have the ability to perform phagocytosis, and they have very, very strong lysosomal enzymes. So these lysosomal enzymes have the ability to completely break down the bacteria. All right? Super, super important. Other things that the Kupffer cells function to do is not just responding to bacteria. I'm telling you, these things are awesome. They can also respond to particular types of toxins. There's special things in bacteria, and like gram-negative bacteria. You have gram-positive and gram-negative. Gram-negative bacteria have a special thing on their outer membrane, which are called lipopolysaccharides. Okay? These lipopolysaccharides can interact with these Kupffer cells. You want to know how? I'll show you how. Let's come down here for a second. Here I'm going to have a Kupffer cell. Let's say here's a Kupffer cell. Okay, there's my Kupffer cell. Here, I'll draw its little nucleus there. Okay? And then what happens is, on its membrane, it has particular proteins, like these protein subunits here. And these proteins are very, very specific. We talked about this again in our actual immune system video, but this is called toll-like receptor type 4. And this toll-like receptor 4 is usually coupled with another protein. And this protein is called CD14. So they call this one CD14. What happens is these toll-like receptors have the ability to identify or bind with these actual endotoxins. And what is this endotoxins here called? It is called lipopolysaccharides. What it can do is it can send signals to the actual Kupffer cell's nucleus. And then the Kupffer cell will start synthesizing a lot of proteins, right? You know that it'll go and transcribe the actual DNA to mRNA. And then mRNA will get translated into various types of proteins. Right? And then what happens? Some of these proteins that it might release are going to control a bunch of different functions. For example, some of these proteins might stimulate specific pathways. So specific here, let's say specific pathways. And it might trigger the release of a bunch of inflammatory cytokines. So it could cause the massive release of a bunch of inflammatory cytokines. For example, just to name a few, it can release what's called tumor necrotic factor alpha. It can release interleukin 1, interleukin 6. Oh, this one is a big one. Reactive oxygen species. That is a huge one. It has the ability to generate a tons of reactive oxygen species. And it also has the ability to release other chemicals like transforming growth factor beta, nitric oxide, and different types of prostaglandins. We can throw that last one in there. Prostaglandins. Bunch of different chemicals, but all of these are inflammatory cytokines and they try to enhance the inflammatory response. So that's really, really interesting. They also have the ability to interact with parasites. So they can also break down parasites. So certain types of parasites, it has the ability, it has special receptors on it that recognize these actual parasites, okay? And the last thing that is really, really important for these Kupffer cells is they have the ability to break down old and defective red blood cells. So for example, let's say I have another Kupffer cell here. Here's another Kupffer cell. If you guys remember, we said that the red blood cells have about 100, 120 days lifespan, right? And as they get older, remember there's a protein called spectrin and ankyrin that control its actual flexibility and pliability. But as the red blood cell gets older, it loses that pliability. And if you remember, there is a very important component inside of this actual red blood cell called hemoglobin. So let's say that this red blood cell is greater than 100 to 120 days. Okay, so it's bye-bye time. This actual Kupffer cell has special enzymes in it that have the ability to break down the hemoglobin. Okay, and what it can do is it can break down the hemoglobin to its two components. One is called heme, and the other one is called globin. Globin is a protein. So what the globin will do is there's going to be special enzymes that will break down the globin into amino acids. But then guess what? We have another component inside of this Kupffer cell. It has another special enzyme that can break down the heme into what's called bilirubin. Eventually it goes to biliverdin and then biliverdin to bilirubin. I'm not going to spend too much time on that. I just want you to know that it actually is broken down into bilirubin. Okay? And one other component of the heme is you release what's called iron. And iron, can actually be bound to specific proteins inside of the cell, which is called apoferritin. And then when it combines with the iron, it forms ferritin, or we can put it into the blood and it combine with transferrin. We've already talked about this many times, right? But the significance here is that bilirubin, we can push it out of this cell, and guess where that actual bilirubin can go? For example, let's say that it breaks down the red blood cell right there, and it pushes out this substance called bilirubin. That bilirubin is actually going to be taken up. There's special transporters located on the actual basolateral cell membrane of this hepatocyte that can take the bilirubin up, and then it can conjugate it. We'll talk about that in the biliary system video, but then that bilirubin that we actually get inside of the cell, we can push it into the actual biliary canaliculi. And then guess what? If we push it into the biliary canaliculi, guess what will happen? It'll go into the bile duct as an important component of bile. Okay, so that's important. Bilirubin is an important component of our bile. Okay, so what have we understood so far? We know the portal venual, we know what it's doing, we know the hepatic arterial, we know what kind of capillaries are coming through here, we know the general functions of this Kupffer cell, okay? So just general ones. Controls phagocytosis of bacteria, it can actually recognize foreign parasites, it can react with different toxins like lipopolysaccharides, and it can actually break down old and defective red blood cells. And it also has an important function with controlling what's called immunotolerance, okay? In other words, it can actually inhibit specific CD8 cells or cytotoxic T cells from completely undergoing specific immunological reactions inside of this tissue. Okay, that's important. All right, one last cell that I want to talk about here is this guy right here, this orange cell. We said stellate cells. They have a particular function too. Son of a gun. That particular function of these stellate cells is they are what's called, they store vitamin A, okay? So they store vitamin A and some fat. So they can store what's called vitamin A. Now, these cells are pretty much quiescent. In other words, they don't really have any specific functions. They don't really do anything crazy. They just are responsible for storing some vitamin A and some fat. But if over time there is consistent, let's say, bacteria or parasites or alcoholic-induced damage to the liver or hepatitis or rare diseases like Wilson's and hemochromatosis, whatever it might be, the liver undergoes consistent damage and inflammation. What will happen is, due to that, these stellate cells can become activated. And what they can do is, so let's say here I have a stellate cell. Okay, they're generally pretty much quiescent, all right? So they're pretty much in just like a resting phase. They're not really doing anything. But if for whatever reason you continue to cause this stellate cell to have a lot of tissue injury, so there's this excessive tissue injury, okay? Over time, again, like hepatitis, cirrhosis, maybe due to hemochromatosis, Wilson's disease, infections, whatever it might be, it can convert these stellate cells into these special cells. And these cells are responsible for producing a lot of what's called collagen type 1. So these are called myofibroblasts. So the satellite cells can become myofibroblasts and produce a lot of collagen type 1. You know why that's important? Because if I start producing a lot of collagen type 1 in this area, guess what it's going to do to the blood vessels? It's going to cause the blood vessels to become really fibrotic. If the blood vessels become very, very fibrotic, it can narrow the blood vessel walls. And then the blood trying to come through that area is going to be impeded. Guess what? The blood's going to backflow. And if it backflows and backflows and backflows, guess what it can produce? Portal hypertension. So that's important because these stellate cells are responsible. They're one of the main culprits of causing this fibrosis of the liver due to consistent and chronic tissue injury. All right, cool. That's that guy. Now, next thing I need to talk about here. You have your hepatocytes, right? We've talked about all these parts here. One more thing I have to talk about is these actual hepatocytes. These hepatocytes are so darn cool. We're going to look at their function later. But for right now, we have to talk about this last thing, which is the bile duct. The actual hepatocytes are responsible for making bile. And here's what I want you guys to remember. If you don't remember anything else about bile, just please remember the three main components of bile. One is your bile salts or your bile acids, okay? So bile salts can come in three different ways. They can be a bile salt, they can be a bile acid, or they can be what's called a conjugated bile salt. We'll talk about that when we get to the biliary system. And this is like cholic acid, chenodeoxycholic acid, and it can combine with taurine and glycine and stuff like that. Another component, this is like 70% of it. This is the most important one. Another one is called phospholipids or lecithin. This is another really important component. And then another important component is called bilirubin. This is what causes the pigmentation in the bile. And there's other components in it too, but these are the big ones. Also cholesterol. The liver cells, the hepatocytes, are responsible for making that bile. So on one side, look at this cell. It's so cool. It's got like a double-sided cell, double-sided, right? One side faces the actual sinusoidal capillaries. And again, this is called the space of Dissy. Here's what's odd. This side has microvilli. Okay, if you look at this, what are these like little things here called? These are called microvilli. They're responsible for increasing the surface area of these liver cells. Okay, super important cells. They're responsible for increasing the surface area. This side where the microvilli is, like imagine here I take a cell. Here's a hepatocyte. On one side it has the microvilli. This side, it's so weird. This is the basolateral membrane. That's what's so odd about this. It's on the opposite side. Usually the basolateral membrane would be this side, but it's different, right, for the actual liver. So on this side is where the actual capillary system is. On the other side is the actual biliary canaliculi. And this side right here, facing that, is called your apical membrane. It's pretty weird. because it's kind of opposite of what you would normally think. Usually we think the apical membrane is going to be towards the sinusoidal capillaries and the basolateral will be towards the bile canaliculi, but it's not. One other thing I want to make sure I clarify here is when you look at these hepatocytes, really the bile canaliculi is actually formed by these two hepatocytes. So imagine here I have an hepatocyte and I kind of make like a little thing like this and a little thing like this. You see how they're two connected? They're actually connected by important structures. 3 important structures actually make up the hepatocyte connections. Tight junctions, desmosomes, and even some gap junctions, okay? But there's this tiny little structure between the two juxtaposed cells. When their apical membranes fuse, it forms these little canaliculi, and that's called the biliary canaliculi. So really, what happens is in between these hepatocytes, they're actually taking all the bile and secreting it into this. But it's easier to show it like this, all right? When we're looking at it in a functional diagram. So really understand that the bile canaliculi is actually formed by 2 juxtaposed hepatocytes. Their apical membranes are fused, but it forms this tiny little groove or tiny little space called the biliary canaliculi. All right, and then again, what happens to this bile? The bile is going to move through this biliary canalicular system and then move out and into the bile duct. And then if you remember the bile ducts will become your left and right hepatic ducts. Those will form the common hepatic ducts. They'll fuse with the cystic duct of the gallbladder and form the common bile duct, which will go down and fuse with the main pancreatic duct. All right, so one last thing before we look at the different types of liver lobules is we said we have this space called the space of Disse, right? And this is where a lot of the exchange between the plasma of the sinusoidal capillaries and the interaction between the hepatocytes are, right? So you know that hepatocytes, they could be secreting different things, and that could be moving into the blood. Or there could be solutes coming from the actual blood and moving to the hepatocytes. But there's this exchange. One last thing is we said that the basolateral membrane is the microvilli, right? It's where the microvilli are. What's really interesting is that there's a lot of connective tissue in this area, in the space of Disse, a lot of connective tissue. So you can say that the hepatocytes, they rest on this connective tissue matrix in the space of Disse. And this connective tissue matrix is made up of a ton of different types of collagen. Okay, for example, it could be made up of collagen 1. It could be made up of collagen 4. It could be made up of collagen 5. It could be made up of collagen type 6, whatever. There's a lot of other proteins too. There's fibronectin, undulin, proteoglycan, so many different structures. But the significance is to remember there's like this connective tissue bed that the basolateral membrane of the hepatocytes rest on, kind of like a basement membrane in a way, which is really interesting. Okay, so now we've really dived into this actual liver lobule. I think now we can safely talk about the 3 different forms of liver lobules at this last part over here. Okay, so last thing we got to talk about here, guys, is there's 3 different ways of looking at a liver lobule. So we looked at a liver lobule in pretty good detail. All right, we talked a lot about it. But there's ways that you can kind of like functionally classify these liver lobules. So let me write that down here for you. There's 3 ways, okay? So one way that we're going to look at is we're going to look at what's called the classic liver lobule, okay? The classic liver lobule. And that's the one that we kind of focused on primarily over there. Another kind is what's referred to as the portal lobule. And then the last one, which is my personal favorite, is the acinus lobule, or the acinus lobule. Here, let's do it like that. Acinus lobule. Okay, so these are just different ways of looking at the liver lobule in a functional way, okay? So let me explain first off the classical one, but that's the easy one. This one is referring to the blood flow. Okay, this one is referring to the blood flow. So it's looking at the blood flow of 2 vessels, okay? The hepatic, arterial, and the portal venial. It's looking at how that blood is flowing from these guys toward the central vein. Okay, so it's looking at the blood flow of the hepatic arterial and the portal venial to the central vein. So when we talk about this way, for example, what does this mean? Okay, here's your portal triad. Here's your central vein. From this, the blood from the hepatic arterial and the hepatic venule is moving in a direction toward the central vein. So to simplify it in one arrow, this blood will be flowing from the portal triad towards the actual central vein. Portal triad, central vein, portal triad to central vein, and vice versa. So the big significance here, when you're looking at a classic liver lobule, is you're looking at the blood flow coming from the portal triad to the central vein. The next one is the portal lobule. And the portal lobule is referring to the actual direction of the biliary flow, okay? So it's looking at the direction of the bile flow. So what do I mean? If you look here, you have to kind of make this a three-way structure. So here, I'm going to connect this central vein and this central vein and this central vein all together in one. So I'm going to come here to this, go to this, and go to this. So what you do is you make like this triangle. Now, in the center of the triangle, it should tell you that is going to be where the actual bile duct is. So now, from each one of these central veins, follow the flow of bile towards the portal triad. So now look, the bile will be flowing this way. The bile will be flowing this way, and the bile will be flowing this way. And in the center is where you should find your bile duct. Okay? That's the significance of this portal lobule, is that the portal lobule tells us the direction of the flow of bile moving from the central vein towards the actual portal triad. Okay? That's that one. The last one, my personal favorite here, is the acinar lobule. And this tells you the flow of arterial blood. So this is going to tell you the flow of oxygen-rich blood. So this one is really darn cool. So what I'm going to do is, let me do this one in black. What I have to do is, I have to take from this central vein, okay? I'm going to go to that point of the lobule. Then from that, I'm going to go to this point of the lobule. And I'm going to do the same thing over here. I'm going to work to this point, and I'm going to work to this point. Now, if you guys remember, there should be blood moving from the hepatic arterial and moving towards the central vein, right? So for example, it would be going like this, and it would be going like this. So the blood is moving from this actual portal triad and moving towards the central vein. It creates what's called this zoning. So now what I'm going to do is I'm going to come down with the baby blue, and I'm going to zone off each area. So this zone right here, this zone right there, and I'm going to come back over here. I'll have this zone right here, that one, and that one. So now look what I form. I kind of form like this rhomboid like structure here. Each one of these is an individual zone. So for example, this will be zone 1. The one closest, or what's called periportal. So the hepatocytes in this area is going to be periportal. They're close to or next to the portal triad. Then we're going to go in the center of this one, and that's going to be zone 2. And as you know now, the one that's closest to the central vein is going to be zone 3. Okay? So this is called the acinus model, or the acinus lobule, is that you're looking at the flow of blood, the flow of oxygen-rich blood from the portal triad towards the central vein. And again, the hepatocytes in this area are referred to as periportal. These are just going to be the ones between. And this is going to be called perivenular, okay? Perivenular hepatocytes. Why did I mention this? Here's what's cool about this one. In each zone, Okay, for example, let's say I have zone 1 particularly and zone 3. Zone 2 is the intermediate between, so we're not going to focus a lot on that. We're just going to say that it does a mixture of functions between these two zones. Zone 1, if you think about it, it's closest to the hepatic arterial. So it's going to get the highest, it's going to get the blood that's richest in the amount of oxygen first. So as the blood is coming through, it's going to have a lot of oxygen. It's flowing through. I'm going to give off oxygen to this cell and this cell and this cell. And I'm going to keep going. Give off oxygen to this cell, and keep going. By the time the blood gets from the actual portal triad all the way towards the central vein, what do you expect the partial pressure of oxygen to be at that point? Lower. So the partial pressure of oxygen in zone 1, we expect to be the highest. And the partial pressure of oxygen in zone 2, we expect to be the lowest. So why is that important? Zone 1 is going to be the most resistant to any type of circulatory compromise. If for any reason your blood is, you have a decrease in blood volume like hypovolemic shock or whatever, this zone is going to be the most resistant because it's going to get the most oxygen from the actual arterial because it's the first one there. Whereas zone 3 is going to get the least amount of oxygen, so there's going to be most necrosis or most death or most ischemia to the cells around the central vein. That's important. Another reason why this is important is because each one of these zones perform different functions. That's one of the coolest things ever because they call it zonal heterogeneity. Heterogeneity. What that means is each zone performs a specific metabolic function. What do I mean? For example, zone 1, it's highest in oxygen, so it's going to want to perform oxidative metabolic pathways. So for example, in zone 1, it's going to have the most oxygen, so you want it to perform a lot of oxidative metabolism. For example, some of the pathways that you'd want it to be is like amino acid metabolism. That could be one pathway there. That's a metabolic pathway that requires the presence of oxygen. Other ones could be gluconeogenesis. That one is also an oxidative pathway that requires, again, it's going to require oxygen, ATP. Other ones would be like cholesterol synthesis, bile salt synthesis, ureagenesis, so urea cycle, so the ureagenesis. Okay, things like that. It's going to be performing a lot of these pathways. Another one is it will perform beta oxidation. So you have another one there which is called beta oxidation. The whole point I want you guys to understand is that if you look at the mRNA that's expressed in zone 1, it's going to be different for the particular enzymes that function primarily in that zone. Whereas if you look at it in zone 3, the mRNA might be more specific to different pathways. That's what's so cool. They actually did studies in zone 3 has the highest mRNA expression of an enzyme called glutamine synthetase. So what do you think that means? That means glutamine synthesis is going to occur most in zone 3. That's the significance here. So this one is going to perform more pathways like this will perform glycolysis because it's not going to get a lot of oxygen, obviously. It'll perform things like fatty acid synthesis. It'll perform things like ketogenesis. Again, it'll perform what's called glutamine synthesis. And another really important one that we're going to talk about in the hepatocyte video is it performs what's called, we'll call biotransformation or xenobiotic metabolism. And that's basically metabolizing different types of toxins and drugs, and whether they be endogenous or exogenous compounds. So that's really, really important to understand. Okay, so 3 different ways that we can functionally classify these liver lobules as classic, which is just saying the blood flow going from the portal triad towards the actual central vein, both the mixture of hepatic arterial and portal venular blood. The portal lobule, which gives us the direction of the biliary flow, which is going to be, if we can look at it in this triangular-like way, it's going to be flowing from the central vein towards the portal triad. And the last one is the ascender lobule, which is making this rhomboid-like structure, and it's separating the hepatocytes into 3 zones. Zone 1, Zone 2, and Zone 3. Zone 1, highest resistance to circulatory compromise because it's going to get the most oxygen. Zone 3, it's unfortunately going to be the one that might have the most necrosis and ischemia and cell death, and because it's not getting a lot of oxygen. Zone 3 will perform pathways like glycolysis, fatty acid synthesis, ketogenesis, glutamine synthesis, xenobiotic metabolism, whereas zone number 1 might be more for amino acid metabolism, gluconeogenesis, cholesterol synthesis, bile synthesis, the ureagenesis, other different pathways. And again, that should make sense because different areas have this thing called zonal heterogeneity. All right, engineers, so in this video we talked a lot about the histology of the liver. I really do hope it made sense. I know it was long, and I thank you guys for sticking in there, and I hope that you guys really did learn a lot, and I really hope it made sense. I truly, truly do. That really is our goal here at Engineered Science, and we're dedicated to that goal. If you guys did like this video, please hit that like button, comment down in the comment section, and please subscribe. Also, if you guys get a chance, go check out our Facebook, our Instagram, maybe even our Patreon account if you guys have the opportunity to donate, maybe even $1. It would make the difference. We appreciate you guys, and as always, Ninja Nerds, until next time.